This is Sandhya, your botany lecturer. In the last video, we have discussed about the biological classification. In biological classification, we have discussed about the two kingdom classification, then five kingdom classification by R.H. Whittaker. In five kingdom classification, five kingdoms are there. The living organisms are classified into five kingdoms: Monera, Protista, Fungi, Plantae, and Animalia. Then about Monera and in detail about Monera and Protista we have studied. In this lecture, in this video, we are going to deal with Fungi, Plantae, and Animalia. First, coming on to fungi. Fungi are the heterotrophic organisms. Heterotrophic organisms are those organisms which are going to depend on other organisms. They can be parasitic or can be saprophytic. Here, this fungi is going to show great diversity in morphology and also in habitat. They are going to present in bread, mushrooms which are edible, and toadstools are there. And some are parasitic fungi like albugo and when the bread is kept like that, it is going to get rotten or fungus grows on it. So the great diversity is in morphology and habitat. Then they are also going to cause some diseases like rust and smut in plants. And majorly these fungi are multicellular, but unicellular is yeast, and this yeast is used to make beer and bread. Then these fungi are also the sources of antibiotics, like penicillium is penicillium for penicillin. Streptomyces, penicillium is for penicillin. So antibiotics are also obtained from this fungi. Streptomyces is a different one. It is an actinomycetes, so it's not streptomyces, but penicillium. Penicillium is a fungi. From penicillium, we are going to get the penicillin antibiotic. Then we are going to keep the food in refrigerator. What for? To preserve the food materials. To preserve the food materials such that the fungi doesn't grow on the food materials because of the low temperature. What happens? The enzymatic activity is retarded, and then the bad bacteria and bad fungus is not going to go in that food. So that's why we are going to keep the food in the refrigerator. So this is about the diverse nature of the habitat and morphology of fungi. Then these fungi are majorly multicellular, except yeast. They are filamentous. They are going to have long, slender thread-like structures called as hyphae. They are going to have long thread-like structures called hyphae. The network of hyphae is called as mycelium. Okay, mycelium. Sometimes this particular hyphae is continuous tubes where multinucleated cytoplasm is present. Such type of hyphae is called as cenocytic hyphae. Where the hyphae are continuous tubes and have multinucleated cytoplasm. So cenocytic hyphae. But in some fungi, they are branched and false septum is present. The septate walls are present. So hence such type of fungi are called as branched and septate. Then in the cell walls of the fungi, chitin is present. Whereas in plants and other things, cellulose is present. Here in the cell walls, the chitin is present, and glycogen is the reserved food material. Then the cell wall is made up of chitin and also it's a polysaccharide, and amino acid is also present. So cell wall is made up of chitin. The reserved food material is glycogen and fat. Coming on to the heterotrophs. These fungi are heterotrophs, isn't it? That means they are majorly heterotrophic. They are heterotrophic, and they are both saprophytic and parasitic are present. Saprophytic, these fungi are going to live on the dead and decaying organisms. Parasitic, they are going to live on the living plants, live plants and animals. So parasitic. And sometimes they are also symbionts. Symbionts, as you can see in lichens. lichen is an organism where it is a symbiotic association of algae and fungi, both algae and fungi. So symbiotic association is there. They are parasitic, and they are also going to show mycorrhiza. Mycorrhiza means these fungi are going to live in association with the roots of some higher plants, roots of some higher plants for nitrogen fixation, like that. So they are saprophytic. Fungi are saprophytic, parasitic, symbiotic. They are symbiotic. Then mycorrhiza also, they are present with the mycorrhiza means they are present in association with the roots of higher plants. This is about the heterotrophic nutrition. Coming on to the reproduction part. Reproduction takes place by vegetative reproduction. Vegetative reproduction, fragmentation. Vegetatively, it is going to reproduce through fragmentation. Then asexual reproduction. Asexual reproduction is also going to take place through conidia, then sporangiospores, like that. Sporangiospores or zoospores. So asexual reproduction is also going to take place through conidia, conidiospores, or sporangiospores, or zoospores. And sexual reproduction is also going to take place by oospores, then ascospores and basidiospores. So these spores, sexual spores, oospores, ascospores and basidiospores are going to form in the fruiting bodies. They are going to form on the fruiting bodies. Then during the formation of these particular spores, some stages are there. Various stages are going to observe in the fungi. The first stage is plasmogamy. Plasmogamy is fusion of two protoplasms of motile or non-motile gametes. Fusion of the protoplasms of motile or non-motile gametes. That is plasmogamy. Then karyogamy. Karyogamy means fusion of two nuclei. Karyo means nucleus, gamy means fusion. Fusion of two nuclei is karyogamy. Then meiosis is going to occur. Meiosis is nothing but the reduction division for the formation of the haploid spores. Meiosis is going to occur. So these are the phases. These are the divisions which are going to occur. Cycles which are going to occur in the life cycle of the fungi. Then so generally sexual reproduction means fusion of male and female gametes is sexual reproduction. So then what happens by the fusion of the two haploid gametes, fusion of two gametes, what happens? The diploid cells are formed. In that case, the diploid cells are formed. So this is general case. But in some fungi like Ascomycetes and Basidiomycetes, what happens? A middle stage is going. Another phase is going to occur. It is called as dikaryon stage. Dikaryon stage. In this stage, what happens? Two nuclei are present are in one cell. Two nuclei are going to present in one cell, and this type of condition is called as dikaryon, and the phase is called as dikaryophase. It's an intervening dikaryotic stage. Then after that, what happens? The two parental nuclei will fuse together and form into diploid cells. That happens. But an intervening stage is going to occur in Ascomycetes and Basidiomycetes. That intervening stage is dikaryon, where two nuclei are present in one cell. So this is about the sexual reproduction. This is the general characteristics of fungi. We have studied about the habitat where they are present. What are the different types of fungi? Then what is the morphological structure? And then mode of nutrition, saprophytes, parasites, symbionts, then mycorrhiza, like that. Then sexual reproduction wise, vegetarian reproduction by fragmentation, asexual reproduction by these spores, sexual reproduction by these spores, and different cycles also we have discussed. These are the general characteristics of the fungi. Then, based on the morphology of the mycelium, mode of spore formation, based on the morphology of mycelium, what is mycelium? Mycelium are the slender filamentous structures. The slender filamentous structures are hyphae. The network of hyphae is called as mycelium. The slender filamentous continuous tubes are nothing but the hyphae. That network, they form into a network called as mycelium. So based on the morphology of mycelium, then mode of formation of gametes, gametes and formation of fruiting bodies. Based on the formation of fruiting bodies also. What are fruiting bodies? I am going to tell later. In this. Then based on these three features, this fungi is divided into 4 classes. The first is Phycomycetes. The second one is Ascomycetes. Third one is Basidiomycetes. Then fourth one is Deuteromycetes. Mycetes means fungi. Okay, mycetes means fungi. So Phycomycetes. So based on morphology of the mycelium, mode of spore formation and formation of the fruiting bodies, based on that fungi are divided into 4 classes: Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. First we'll discuss about the Phycomycetes. This Phycomycetes is also called as algal fungi. It's also called as algal fungi. They are mostly aquatic. They live in water. They are of aquatic habit. They are also present on decaying wood and moist damp places. These Phycomycetes are present. They are also called as algal fungi. And the mycelium is aseptate and cenocytic. Aseptate means the septa are not present. The walls are not present inside the mycelium, inside the hyphae. And cenocytic. Cenocytic means many nuclei, multinucleated cytoplasm is present. So the mycelium is aseptate and cenocytic. Then asexual reproduction is going to take place by 2 types of spores. Zoospores, which are motile in the sense they can move. Then aplanospores. Aplanospores are non-motile spores. They cannot move. So asexual reproduction is going to take place by zoospores and aplanospores. Then these spores are going to be present in the fruiting body, sporangium. Then sexual reproduction is going to take place by zygospores. Zygospores are there, and they are formed by fusion of two gametes, and these 2 gametes can be similar. If they are similar, they are called as isogamous. If they are similar, isogamous. If they are similar, if the gametes are similar, isogamous. If the gametes are dissimilar, not same in structure, dissimilar, anisogametes. So zygospores are formed by the fusion of the gametes, which can be isogamous or which can be anisogamous. Then very good examples of Phycomycetes are Rhizopus, which is also called as bread mold. Why it is called as bread mold? When the bread is kept like that for few days, what happens? It gets spoiled. Spoiled in the sense the fungus grows on that. The fungus which grows on that bread is nothing but the rhizopus. Then mucor. Then albugo. Albugo is a fungi. It is a parasitic fungi which grows on mustard. It is parasitic which grows on mustard. That's about Phycomycetes. Now moving on to single fungi. Coming on to Ascomycetes. It is called as sac fungi. It is called as sac fungi. In this Ascomycetes, yeast is going to come. Yeast is an example. Yeast is unicellular. It is useful for the formation of bread and also for beer. So yeast is yeast. What's the scientific name? Saccharomyces. Saccharomyces. It is unicellular. Then multicellular is penicillium. Penicillium, where the penicillin antibiotic is extracted. Then here also they are mostly saprophytic. They are mostly saprophytic, decomposers, and coprophilous. Saprophytic, decompose, and coprophilous. Coprophilous in the sense they are going to live on the cow dung, the fecal matter of the animals. So growing on the dung. The mycelium is branched and septate, and asexual spores are called as conidia. The asexual spores are called as conidia. So these spores are going to form on a special hyphae called as these are formed on a special hyphae called as conidiophore. These are formed on a special hyphae called as conidiophore. Conidiophore. Then this conidia are going to germinate and form into new hyphae. This is asexual reproduction. So asexual reproduction by conidia. Coming on to sexual reproduction. Sexual reproduction is going to form by ascospores. Ascomycetes, no, ascospores. Ascospores. And these ascospores are formed on asci. Asci. They are going to form on a asci. They are formed endogenously in asci. This particular asci, this particular asci are arranged on ascocarps. So in this way also you can say, see, this particular Ascomycetes is going to have the fruiting bodies ascocarps, which bear the asci, which produces the ascospores, like that. Then these ascocarps are the fruiting bodies. Then these ascocarps are of three types. If that is flask shaped and without any opening, it is called as clistothecium. If it is flask shaped and without any opening, it is called as globose shape, not flask shape, sorry. Globose shape without any opening is called as clistothecium. Then if it is flask shape with an apical opening, that is called as perithecium. It is flask shaped. Clistothecium is globose shaped, okay? Globose shaped. Perithecium is flask shaped with an opening. This globose shaped clistothecium does not have any opening. Perithecium is flask shaped and it has an apical opening. Then apothecium, another form is apothecium. Another form is apothecium. It is cup or saucer shaped with a saucer shaped. Cup or saucer shaped ascocarp. Saucer shaped ascocarp. These are the different ascocarps. Then examples of Ascomycetes are Aspergillus, claviceps, then penicillium, and neurospora. Penicillium and neurospora. This neurospora is widely used in the genetic studies and in the biochemical work. This particular neurospora is widely used. Then many members like morels and truffles are edible. They can be eaten and mada. Many members of morels and truffles are edible and are considered as delicacies. And they belong to Ascomycetes. This is about Ascomycetes. Coming on to Basidiomycetes now. Coming on to the Basidiomycetes. Basidiocarp. Ascomycetes, ascocarps. Basidiomycetes. They are also called as club fungi. They are also known as club fungi. And in this Basidiomycetes, the mushrooms come, then bracket fungi or puffballs are included in this Basidiomycetes. Puffballs are included under Basidiomycetes. They grow in soil, logs, and in tree stumps, living in both plant bodies as parasites also. And when they are living as parasites in plant bodies, they majorly cause two diseases: rust and smuts. So rust and smuts are also nothing but they are fungi belonging to Basidiomycetes. Then asexual spores are generally not found in this, but vegetative reproduction takes place by fragmentation. Then here the sex organs are absent. Okay, so asexual spores are not found. Then vegetative reproduction by fragmentation. Then so sex organs are absent, and here the plasmogamy is going to be observed, where the fusion of two protoplasms of two gametes are going to occur. Plasmogamy is there. Then this plasmogamy is brought about by fusion of two vegetative or somatic cells of primary mycelium or of different strains or genotype. Then what happens? An intervening stage is going to occur, which is called as dikaryon. As we have studied, dikaryon. It is nothing but two nuclei in one cell. Then karyogamy and mesogamy are going to occur in the basidium. Okay, basidium. So basidium is going to produce basidiospores. How they are going to be produced? By the karyogamy and meiosis. It produces basidiospores. These basidium are arranged in basidiocarps. The fruiting bodies are nothing but basidiocarps. So in the basidiocarps, basidium are present. The basidium are going to undergo karyogamy and meiosis to produce the basidiospores. The very good examples of this is nothing but agaricus. Agaricus, which is mushroom. Okay, we have studied. Now mushrooms are there. Then Eustiligo, it's a smut. Puccinia, it's a rust. Polyporus, it's a bracket fungi. Then Lycopterodon is a puffball. Okay, agaricus mushroom is just smut. Puccinia rust, Polyporus bracket fungi, Lycopterodon puffball. These are the examples of Basidiomycetes. Moving on to the fourth class, which is Deuteromycetes. Okay, Deuteromycetes is also called as imperfect fungi. It is also called as imperfect fungi because only the asexual or vegetative phases of these fungi are known. Only the asexual and vegetative phases of these are known, and sexual forms of these fungi were discovered and they were moved into classes. They rightly belong to it. So they are imperfect fungi, madam. Imperfect fungi. Then they are present. They are present in. They are having asexual spores called as conidia. So you can say the vegetative vegetative phases and the asexual phases are known. When the sexual phases are they're discovered, they were moved on to another classes like Ascomycetes or Pseudomycetes. But whatever the vegetative and the asexual phases are known, they are included in Dendromycetes. But when the detailed account of sexual phases are also discovered, they are moved on to Ascomycetes or Pseudomycetes. So asexual spores are Conidia. They are branched in septate, same as that of Pseudomycetes and Ascomycetes. Then they are present as parasites, and they are also present as decomposers of litter, which are useful for recycling of the minerals. Very good example is Alternaria. Alternaria, and which is called as early blight. Then Coletotrichum, which causes red rot of sugarcane. So if we open the sugarcane, if we do the longitudinal section of the sugarcane, if we observe a long strip of red color one, it is nothing but the red rot, which is caused by the fungi, which belongs to Dendromycetes. That fungi name is Coletotrichum. Then Trichoderma. These are the examples of Dendromycetes. Okay. Coming on to, this is about the third kingdom, fungi. Coming on to the fourth kingdom, Plantae. Plantae are eukaryotic. And chlorophyll bearing organisms. Bearing organisms and commonly called as plants. And we can also say that these are autotrophic. Majorly they are autotrophic, but few plants are there which are carnivorous, which are insectivorous, which are parasitic. So insectivorous or carnivorous plants are like Nepenthes, bladderwort, Venus flytrap. These are insectivorous plants. Means for some nutrients like nitrogen, insectivorous or carnivorous plants. These are heterotrophs. They are insectivorous and carnivorous plants. As they grow in nitrogen deficient soils, for nitrogen requirement, they trap the insects. They consume the insects and get the nitrogen requirement. Then also parasites. Parasites are also there in plants, like parasites like Cuscuta, Viscum, Striga. So these are all plants which live as parasites inside the plants. These are plants which live in plants anamata. They depend on other plants for the nutrition, or for other requirements. And they are eukaryotic and they have chloroplast. And the cell wall is made up of cellulose. And a prominent, well developed and a prominent chloroplast are present. That's why these are autotrophs, and they are going to prepare their own food materials. Then the reserve food material is starch. Then, and they show absorptive mode of nutrition. Then these are again divided into algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Okay, these plants are divided into algae, bryophytes, pteridophytes, and gymnosperms and angiosperms. In detail, we are going to study in the next video, next chapters, based on this. Then coming on to the next kingdom, Animalia. Coming on to next kingdom, Animalia. Animalia are also eukaryotic and heterotrophic. Eukaryotic and heterotrophic, and they're directly and indirectly dependent on plants, and they digest their food in a cavity. They digest their food in a cavity, and the reserved food material is glycogen or fat. Glycogen or fat, and their mode of nutrition is holozoic. It means the entire food is taken. The entire food is taken. The ingested food is taken. By ingestion of food, there is nothing but holozoic. Then they follow a definite growth pattern. Like in juvenile stage, in the early stage, the growth will be there. Then after the adult stage, the growth stops. It's definite. They acquire a particular shape and structure. And the higher forms are having elaborate sensory and neuromotor mechanism is there. Sensory and neuromotor mechanisms are there, and most of them are capable of locomotion. They can move. They are capable of locomotion. And the sexual reproduction is by copulation, the fusion of male and female gametes, followed by the embryonical development. So in detail and all about the classification we are going to study in the zoology subject. So that is about the 5 kingdom classification, which was given by R.H. Whittaker. But still some flaws were there. Then after 5 kingdom classification, a 6 kingdom classification was given by Carl Woese. Carl Woese gave 6 kingdom classification. 6 kingdom classification, where the kingdom Monera has been replaced with 2 kingdoms. It is bacteria and archaebacteria. Bacteria, as usual then Protista, Fungi, Plantae and Animalia. The difference is that the Monera kingdom has been replaced with 2 kingdoms, bacteria and archaebacteria. And here, using a small subunit of ribosomal RNA, that is 60S rRNA as a marker, small subunit of ribosomal RNA as a marker. These organisms were studied for their evolution relationships. And Carl Woese and co-workers have found out that there are some close relationships are present, and we can say that they can be divided into three categories. Three main categories, or we can call them as domains. It is bacteria, archaebacteria and eukarya. So these living organisms can be divided into 3 main domains. There is bacteria, archaebacteria, you can say. Archaea and eukarya, and here these domains are having common ancestral group, common relationship, common identity of ancestors, of early living organisms, which are called as progenotic. Are having common ancestral group of early living organisms, progenotic. Then archaea and eukarya are closely related than that of bacteria. This was done by Carl Woese 6 kingdom classification. Then after this, the 5 kingdom classification didn't include a few organisms such as viruses, viroids, prions and lichens. They didn't include. Why? Because it did not fall into any of the kingdoms. So So let us learn about those organisms which do not fall into any of the kingdoms. Exceptional organisms. First is viruses. Viruses are acellular. They are non-cellular structures. They are acellular, non-cellular structures, inert crystalline structures when they are present outside the living body, outside the living cell, Then these are obligate parasites. They are obligate parasites when they are present outside the living cell. They are present in the form of inert crystalline structures, inert crystalline structure. But when they infect a living cell, when they infect a living cell, they are going to take over the machinery of the host cell, and they infect that particular host cell, replicate themselves, and kill the host. So they are obligate parasites. They do not have any cell structure. They are non-cellular, acellular structures. So, and the name virus means poisonous. The name virus means poisonous fluid or venom, which was the name was given by Pasteur, which means poisonous fluid or venom. Then, so as we are saying that they do not have cellular structure, then what do they contain? They contain nucleic acids and proteins, isn't it? The main important structures are present. All other accessory structures are not present. Main important nucleic acid and proteins are present. Nucleic acid, proteins are present. The nucleic acid can be RNA or DNA. RNA or DNA. But known virus is having both RNA and DNA. If they contain only RNA viruses. If they contain only DNA viruses. And the proteins are present in the form of a cover or a coat, which is called as capsid. And the units of capsid are called as capsomeres. Okay, so the viruses are having nucleic acids and proteins. Then tobacco mosaic virus. Tobacco mosaic virus and human immunodeficiency virus. Human immunovirus. Immunovirus, HIV are RNA containing viruses. And there are some viruses which infect the bacteria. They are called as bacteriophages. The viruses which infect the bacteria are called as bacteriophages. They are DNA containing viruses. If we see the structure of tobacco mosaic virus, it is having the capsid is nothing but the protein coat, and it encloses the nucleic acid like this. And the capsid is present like this. Like this. This is the nucleic acid core. This is TMP virus now. So what is the nucleic acid core? RNA. These are capsomeres. These are the subunits of capsid. So all these capsomeres will fuse to form as protein code called as capsid. This is TMP virus. Then if you take the bacteriophage, like this. Okay. this is the bacteriophage. So this is the bacteriophage. This is the neck. This is the tail. These are the base plates. These are the tail fibers. This portion is called as the head, where the DNA is present. This is the tail. This portion is nothing but the neck. These are the tail pins or tail fibers, which are useful for the anchorage to the host anamata. They are useful for the anchorage to the host. Host is bacteria. So this is the bacteriophage. Neck or the collar. You can call it as a collar. This head portion is having the nucleic acid. This is bacteriophage. Means viruses which infect the bacteria and they are having DNA. So they are DNA containing viruses. This is about viruses. Then coming on to next exceptional organism is viroids. Next exceptional organisms are viroids. In 1971, T.O. Diner, he observed a small infectious agent, smaller than the viruses, and that cause a disease called as potato spindle tuber disease. Potato spindle tuber disease. Potato spindle tuber disease, and it was found that it is having only RNA. It is having only RNA, no proteins at all. In viruses, nucleic acids are present and proteins are present. But here in this, only the nucleic acid is present, that too RNA is present, and it is of low molecular weight. This was first discovered by T.O. Diner, and it caused a disease called as potato spindle tuber disease. It is having free RNA. They are smaller than these viroids are smaller than the viruses. Coming on to next exceptional organisms, prions. Prions, they contain only proteins. No nucleic acids at all. These are the infectious agents which contain only proteins but not nucleic acids. Then they are going to cause a disease called as scrapie disease and mad cow disease. Prions are the infectious agents which contain only proteins, not nucleic acids, and they are going to cause two diseases, scrapie disease and mad cow disease. This is about prions. Coming on to next exceptional organisms is lichens. Lichens are different because they are not single organisms. They are the mutual association associative organisms, and symbiotic association organisms. They are having symbiotic association, where the two organisms are going to live mutual understanding, mutual association is present. So what are those two organisms? Algae and fungi. So lichens are symbiotic association, mutually useful associations between algae and fungi. The algae is called as phycobiont. The fungi is called as mycobiont. So I am saying mutual understanding, mutual association. So what are they going to do? This algae is going to prepare the food materials. This fungi is going to provide shelter and absorb the water and mineral salts and water to its partner. So they are having some understanding and water. So they are the association is so close that you cannot say that it's a single, that you cannot say that it is having two organisms. It looks like as if it is a one organism, but this lichen is an association of two organisms. Then lichens, this is very important. Lichens are very good pollution indicators. Very good pollution indicators. Pollution. Lichens are very good pollution indicators. They do not grow in the polluted areas. Lichens are the organisms which are very good pollution indicators, and they do not grow in the polluted areas. The study of lichens is called as lichenology. This completes our chapter, biological classification. Thank you.